An increasing number of healthcare-associated infections are caused by drug-resistant bacteria, resulting in more than 50,000 avoidable deaths in North America and Europe every year, with many more deaths occurring worldwide. The world agrees. We need better diagnostics to quickly detect drug-resistant bacteria so we can prevent their spread. Unfortunately, traditional methods such as culture are labor-intensive and too slow to deliver time-critical information. And more recent methods that detect DNA are faster, but can only detect the specific genetic targets they are designed for. Since bacteria continue to adopt new mechanisms of drug resistance, the number of genetic elements which molecular tests need to target continues to increase. Now, diagnostics based on novel and innovative Smarticles technology can improve how we prevent and treat drug-resistant infections and can work much faster than traditional culture methods. Smarticles technology consists of custom-designed DNA packaged into DNA delivery bioparticles whose outer structures contain proteins that bind to specific receptors on the surface of target bacteria. Once the DNA is injected into the bacteria, it causes live bacteria to produce light. The outer structure of each bioparticle is based off of a naturally occurring bacteriophage. However, these bioparticles are not bacteriophages. While we do borrow the outer structure of the bacteriophages for their binding characteristics to target bacteria, the custom-designed DNA is packaged into the structure instead of the original bacteriophage genetic material. Let's take a closer look at how Smarticles technology works. Step 1. Cell surface binding. When bioparticles based on Smarticles technology are added to a sample, the bioparticles bind directly to target bacteria and not other species of bacteria. Step 2. DNA transfer. Once the bioparticles attach to the target, the custom-designed DNA molecule is injected through the cell wall. Step 3. Reporter production. On this DNA molecule is a specific sequence that encodes bacterial luciferase, a bioluminescent enzyme originally discovered in bacteria. This DNA sequence is noted as LuxAB. Upstream of the LuxAB gene is a promoter, a small sequence of DNA that instructs the cell to express or produce whatever is coded downstream of it, in this case the luciferase protein. Step 4. Substrate addition. Following a period of time allowing for LuxAB transcription and translation, bacterial luciferase accumulates in the target bacterium and will produce light when the appropriate substrate is added. When added to an assay, bacterial luciferase substrates penetrate the bacterial cell wall and react with enzymes, resulting in the production of light. This light production signals that the target bacteria is both present and alive. Smarticles technology can also be used to measure antibiotic resistance or susceptibility by adding antibiotics directly to the assay. Antibiotics work by either killing bacteria or stopping their growth, and they do this by interfering with cellular processes, including DNA replication, RNA transcription, and cell wall synthesis. Bacteria that are exposed to an antibiotic that successfully interferes with these cellular processes will not produce light. These are considered susceptible bacteria. If the antibiotics are not successful in interfering with cellular processes, then the bacterial cells will still produce light. These would be considered resistant. This technology allows us to address the problem of identifying drug-resistant bacteria in just a few hours, directly from a patient sample.